How is that so hard to understand? I'll tell you one reason. They want to make money on the drug, and they have to show that the drug by itself has therapeutic benefit. But if I put, so it's more important to make the money than it is to keep the person alive. This is the bottom line. So people have to know that. I'm more interested in seeing how long I can keep people alive, because that to me tells me we know what we're doing. And it's the use of different drugs, different diets, different procedures that help each person as a unique individual live longer with a stage 4 cancer, or any kind of cancer for that matter. So I'm more interested in keeping people alive than making money. So what is responsible for this incredible increase in cancer? As you said, well, it's never one thing. When you have a disaster like this, or an epidemic of whatever, there's never just one thing that contributes to the crisis, or to the disaster. Let's put it that way. And this is a medical disaster of monumental proportions. We're seeing younger and younger people getting cancer. Let's go through the explanations for why we have an increase, a continued increase in cancer. The obesity epidemic is now replacing smoking as a major risk factor for cancer. Obesity is linked to type 2 diabetes and systemic inflammation in the body. Systemic inflammation damages mitochondria. Mitochondria is the organelle in the cell that's responsible for efficient energy production. Disruption of that organelle's ability to produce energy efficiently is the origin of cancer. When that organelle becomes corrupted in a variety of different ways, from inflammation, from radiation, from chemical carcinogens, from oncogenic viruses, intermittent hypoxia, as I said, these kinds of things, and age by itself, aging. Any one of these provocative agents can interrupt the function of energy efficiency, leading to compensatory glucose and glutamine-driven fermentation and dysregulated cell growth. So that's the origin of cancer. Now, why is it that the numbers keep increasing and the deaths keep increasing? Because the World Cancer Institutes, from the National Cancer Institute in the United States to the NHS in England, to all the major academic and research centers, they all are under the belief that cancer is a genetic disease. And when you take the world power of treatment and focus on something other than what the disease is, you have no successful management. So when people say cancer is a genetic disease, they're focusing on things that are not central to the problem. Most of this stuff is secondary downstream effects. The mutations that we see in cancer, the development of precision medicines to try to target these mutations and the consequences of the mutations, all of this is downstream epiphenomena. If it's not central to the problem, then the problem persists. The problem does not go away. We know that cancer is driven by a fermentation metabolism, that's energy without oxygen, and the fuels for that are the sugar, glucose, and the amino acid glutamine. And we know that the cancer cells cannot burn fatty acids or ketone bodies. So once you realize that all cancers that we have looked at have a problem in generating energy through oxygen and are all dependent to some degree or another on fermentation of two different fuels and cannot burn fatty acids or ketone bodies, then the appropriate approach to managing the disorder is to simultaneously restrict the two fuels driving fermentation and transition the whole body over to a fuel, ketone bodies and fatty acids, that the cancer cells can't use. And then they will gradually be eliminated slowly. And this will drop the death rate and will make cancer a very manageable disease. But the reason why that's not being done is because the field thinks cancer is a genetic disease, and they're not focusing on what I say. And why is that? Because ideological dogma. People have been indoctrinated into thinking cancer is a genetic disease, and any form of indoctrination whether it's political, religious, or scientific, prevents the brain from being rational. You have to have rational thinking to break through dogma. That's the way I look at it. And as long as people are dogmatic in thinking cancer is a genetic disease, the numbers of dead people will continue to increase year in and year out. That pretty much summarizes 100 years of scientific research in this area.
Can cancer cells survive without glucose and only glutamine?
They can survive, but they can't grow. If I take away the glucose, the glutamine will keep them alive to some extent, but they can't grow because they need the carbons from the glucose molecule to grow. If I have glucose and take away the glutamine, the cancer cell can survive, but it can't grow because it needs the nitrogen from the amino acid to grow, to make new DNA and RNA. It's the two fuels together that are absolutely essential for the continued dysregulated growth.
And there is a lot of confusion about glucose. What does glucose do to the mitochondria?
Glucose fuels mitochondria. You and I are burning glucose right now in our mitochondria, okay? But if we had cancer, glucose would be fueling the cancer as well. So, but the cancer cell needs the glucose. Absolutely, it's a requirement. Our normal cells can switch to fatty acids and ketone bodies, which the cancer cell cannot use. So then you can marginalize the ability of that cell to survive by taking away the glucose and also the glutamine. It has to be done together because some cancers require more glucose than glutamine, other cancers more glutamine than glucose. So if I take away both fuels simultaneously and transition the body over to ketosis, nutritional ketosis, then you can kill the cancer cells. But you have to be careful because glutamine is an essential amino acid required for the health of our gut and our immune system. That's why we developed the press pulse therapeutic strategy, which chronically stresses glucose availability, but intermittently targets glutamine. Because kill cancer cell, keep immune system healthy. Kill cancer cell, keep immune cells and the gut healthy. Kill cancer cells, keep the immune. So on, off, on, off, on, off, on, off. It's a pulsing. If you try to chronically target glutamine, you paralyze the immune system and damage the gut. So you have to know basic physiology of all organ systems, how they work together, and the vulnerability of the tumor cell. And based on that understanding, all major cancers can be managed. Whether or not they can be cured, I don't know, but we can certainly manage them more effectively without toxicity. That's important. Glucose is not a carcinogen. People have to realize that. They misunderstand what glucose is. If you have cancer, then glucose becomes a fuel. But glucose is not participatory in the origin of cancer. What glucose can do is it can contribute to diabetes and obesity, which create inflammation in the bloodstream, which then damages oxidative phosphorus. But glucose itself is not a carcinogen. But glucose is, if you have cancer, which is dysregulated cell growth, glucose is the fuel driving dysregulated cell growth. So if you take away glucose, that shuts down a primary fuel for the dysregulated growth, but not the other fuel. You got to target glutamine as well.
And could you tell us why is the metabolic therapy so effective?
Because the cancer cells require the two fuels, the fermentation fuels, and they can't grow, and they can't grow without these two fuels. So they can't use anything else. So metabolic therapy restricts those two fuels, killing many tumor cells and making other parts of treatments much more effective. So without those fuels, you can't make new lipids, proteins, or nucleic acids. You need building blocks. Number one, you need building blocks, and number two, you need energy. And without energy, nothing can grow, nothing can live. How do we know that? Because we know cyanide is a poison. People who take cyanide are dead instantly because cyanide shuts down all the energy in the cell. Okay, you die. Without energy, you die. Cancer cells require energy, but they use an alternative form. They use substrate level phosphorylation, basically a fermentation metabolism, rather than oxidative phosphorylation. And we have found and published that the glutamine can generate energy in the mitochondria using a substrate level phosphorylation or a fermentation metabolism to replace the lost energy from oxidative phosphorylation. Interestingly enough, if a person has a tumor, this was done by Warburg and others, and us too, and give the animal the cyanide, it kills the mouse or the host, but not the tumor. The tumor can live without oxidative phosphorylation. So that tells us it must ferment. What does it ferment? Glucose and glutamine. How do you know this? Because we tested it. We tested all the different amino acids to see which one is the most effective, and glutamine is the most effective. So you target the glutamine and take away the glucose and transition to ketones, the tumor cells die. They can't live, and then you know how to manage the cancer. It's not that complicated. It was made to be complicated, massively complicated, because people thought cancer is a genetic disease. And there's millions of different kinds of mutations, making a bewilderingly complicated disease. But when you look at it based on the energy and bioenergetics, it's a very simple disease. It's driven by fermentation. And then you say, well, if that's known, why don't we target fermentation metabolism in cancer patients and manage the disease? And the answer is because the field thinks cancer is a genetic disease. They're brainwashed into thinking it's something other than what it is. And therefore, we get 1,700 people a day dying needlessly from this, and around the world as well. So, I mean, it's the greatest tragedy in the history of medicine. I just, once people wake up to realize what I am saying, people are going to just be so mortified by the fact that we had a process, we have a way right now to drop this death rate and keep people alive longer, and we are not doing that.
I remember I read a paper where you published where the higher the blood sugar, the faster the tumor grows, and the lower the blood sugar, the slower the tumor grows.
Yes, we were the first, I think, to publish this back in 2002, where we showed a direct relationship between how high blood sugar is and how fast the mouse tumor in the brain would grow. So we showed that the lower the sugar, the slower the growth, and the higher the sugar, glucose, of course, the faster the tumor growth. Then we started to see many of similar kinds of studies done in humans with all kinds of different cancers: breast cancer, colon cancer, brain cancer. The higher the sugar, the more rapidly the tumor would grow. And the lower the sugar, the less rapidly the tumor would grow. And it was published in many, many papers showing this relationship between the level of blood sugar and how fast the tumor would grow. And this is why I don't understand how some oncologists or even physicians would say that glucose has nothing to do with the growth of the tumor. It absolutely does. It's in the literature. A big problem, another reason why we're not making any advances, is the people who should know the information were never told of the information, nor do they read the scientific literature. So that's another reason why we have this catastrophe in cancer, is that people don't read the scientific literature, or even worse, don't even understand it. So, and then we have the tragedy.
What blood sugar level did you observe where the tumor doesn't grow?
That goes to the glucose ketone index calculator. And don't forget, the brain is largely responsible for taking in most of the sugar in our body. Okay, the brain. However, the brain can switch over to ketone bodies when you lower blood sugar. So, but the cancer cell can't switch over. So this is the difference. So many of our heart and muscles and things like this, they can all switch to ketones and fatty acids while you lower blood sugar. But the brain, even when you lower blood sugar, the brain will take what's available before any other organ will. The last group of cells that will get the sugar will be the cancer, because if you exercise, muscle takes sugar out of the bloodstream to build glycogen and burns it in the muscle. The brain takes what's available, and the cancer cells are the last one to take what's left. So even if you lower your blood sugar down to a low GK, glucose ketone index is what we call it. You say, well, you didn't get rid of all your glucose. That's true, because the muscle and the brain need it before the tumor cell needs it. So the last cells to get it are the tumor cells. So when you lower down, the brain and the muscles are going to take what little you have in there, and the tumor cells are going to get much, much less, and they're going to die. So exercise and low GKI puts a tremendous stress on the availability of the tumor cell to survive. And then you simply come in with a drug that targets the glutamine, you slaughter these tumor cells, slaughter them. So, and you keep the body healthy while you're doing this. And gradually the tumor gets smaller and smaller and more indolent, where a surgical procedure or a radiation procedure or some final procedure with an immunotherapy could potentially cure the disease, potentially. But you put the metabolic pressure first, and then you come in with the other strategies to remove the final survivors.
And if someone cannot tolerate ketogenic diet and wants to reach this ketogenic state, what can they eat?
You know, it depends on each person. Some people can't tolerate ketogenic diets, but they can tolerate a carnivore diet or a fish diet. It depends on the person. What is the best way to keep the person relatively happy or not stressed out and killing cancer cells? And everybody has to determine that for themselves. They always ask me, oh, he never said what to eat. I cannot tell you what to eat because you might be different from the other guy. He said to eat that, and I ate that, it didn't work, and he ate that and it worked. Just use your glucose ketone index with the Keto Mojo meter, and you'll know what you can and cannot eat. And that varies from one person to the next. So, as I said, if you have a diet that's balanced appropriately in micro and macro nutrients and low and highly processed carbs, it's probably pretty good for you. Eat a little bit of it and see what it does to your GKI. And then you'll know whether or not you're in the zone of therapeutic ketosis, which is healthy, keeping your mitochondria healthy, reducing oxygen-free radicals, and reducing the risk for cancer and other chronic diseases. Diet can lower glucose, but a diet cannot lower glutamine. Therefore, it's a press, pulse, and the pulsing comes from drugs.
What are some of the agents that lower glutamine, and which one did you find to be the most effective?
Well, you can lower, we actually found that the drug 6-deoxy norlucine, it looks like glutamine. That's the in our research, that's the most effective so far, which targets the ability of glutamine to be metabolized to glutamate. So it blocks that first metabolic step. That drug was in fact used for many years on children and people with different cancers, but it was discontinued because they said it was toxic. It's not as toxic as the red devil, doxorubicin, and all these horrible chemicals. It's off patent, so people can't make money on it. But the other thing too is when they did use DON, which is deoxynorlucine, they never targeted glucose. They did, they didn't do it right. They didn't do it right, and they didn't transition the body to ketones. And therefore, the most important drug that we know of to manage cancer is not being used because they didn't do it right. So, but the drug companies are making other drugs that can target glutamine, and these are coming on the market, but they don't target the glucose and put the body in nutritional ketosis when they're using their drugs. So the efficacy of these drugs can be some efficacy for sure, but they're not going to be maximum unless you target the glucose when the body is in nutritional ketosis. How is that so hard to understand? I'll tell you one reason. They want to make money on the drug, and they have to show that the drug by itself has therapeutic benefit. But if I it's more important to make the money than it is to keep the person alive. This is the bottom line. So people have to know that. I'm more interested in seeing how long I can keep people alive, because that, to me, tells me we know what we're doing. And it's a use of different drugs, different diets, different procedures that help each person as a unique individual live longer with a stage 4 cancer, or any kind of cancer for that matter. So I'm more interested in keeping people alive than making money.
Could you talk to us, please, about the experiment where they took the nucleus from a cancer cells and put it in a normal cell, and took the nucleus from a normal cell and put it in a cancerous cell?
Nuclear mitochondrial transfer experiments
Yes.
were telling us that if you take the nucleus out of the tumor cell, isolate the nucleus from the cytoplasm of a tumor cell, and put that nucleus into the cytoplasm of a normal cell, and then put that cell under the skin of a rat you don't get any growth. You don't whatever happened suppressed whatever was in the nucleus that was thought to cause dysregulated growth. It was reduced, removed. On the other hand, if you take the nucleus of the normal cell and put it into the cytoplasm of a tumor cell, and then put that under the skin of the rat, you got dysregulated growth and a big tumor. Yeah. So clearly, what it was telling us, it is the mitochondria in the cytoplasm, not the mutations in the nucleus, that are causing the dysregulated cell growth. And this has been done both in vitro in culture dishes, as well as in living animals, frogs and mice, all of these things. And then on the other hand, if you take an aggressive tumor and remove the mitochondria from the cytoplasm and put in new mitochondria from a normal cell, you get a suppression of the dysregulated growth. On the other hand, if you have an indolent tumor growing very, very slow, and you remove the mitochondria and put the mitochondria from the aggressive tumor into the cytoplasm of the indolent tumor now becomes super aggressive. So clearly, dysregulated cell growth is due to abnormalities in mitochondria structure and function, not in the mutations in the nucleus.
That supports that it's a metabolic disease rather than a genetic disease.
That's the strongest evidence that we know of. And then we also know that you have to realize in the genetic dogma that you have many thousands of different kinds of mutations that can be found in tumor cells. And not all the mutations are considered drivers of the dysregulated growth. Only some mutations were thought to be drivers. They call driver because that mutation is more important than the passenger mutation, which just doesn't do anything. So what we're now finding, so the genetic field said, oh, cancer is a genetic disease, but only due to driver mutations. So if you have these drivers, that would lead to dysregulated cell growth. But now new information is showing that normal cells in our body, you, me, we have mutations in driver genes that are not causing any dysregulated cell growth. So that's another piece of evidence that completely disengages the gene theory, that we have driver mutations in our body that don't form cancer. And now we have some cancer cells that have no mutations. So yet they're dysregulated in the cell growth. Not too many, very rare, but it does happen. So clearly what this is telling us, the nuclear mitochondrial transfer experiments, the findings of driver gene mutations in normal cells, all of this is telling us the cancer cannot be driven by mutations. Dysregulated cell growth is not caused by somatic mutations. And the germline mutations that are risk factors, all of the germline mutations disrupt mitochondrial energy function. So clearly, origin of cancer it's a mitochondrial metabolic disease. And the mutations that we see in cancer cells come from the abnormal metabolism of the mitochondria. So reactive oxygen species are carcinogenic and mutagenic, and they're coming out of the abnormal mitochondria. And they cause the nuclear gene mutations and broken chromosomes and this kind of stuff. So clearly, when you put all the parts together, cancer is a mitochondrial metabolic disease driven by a fermentation metabolism involving glucose and glutamine and an inability to transition over to fatty acids and ketone bodies. How do we know that? I did the experiments. I showed the evidence and published the evidence in the scientific literature. And then you say, well, how come nobody wants to? Because they're dogma. The dogma. The brain can't think when it's indoctrinated. And the jobs and the money and the billions and billions of dollars locked into a failed theory. And we have 1,700 people in the United States dying every day because the brain can't get out of the dogmatic view. It's an ideological dogma. And I tell you this, I show you all the evidence. I did the experiments. I show the efficacy. Can't be true. Can't be true. You tell me what's wrong with that picture. You tell me what's wrong with this picture.
I mean, it's difficult to argue against anything you said, and you have explained it very clearly. Do you have this sort of conversation with oncologists, and what is their reaction?
They get pissed off. A lot of them get angry. They get angry because they said, I never heard this and never taught this in medical school. Oh, because you haven't heard it in medical school, that means it can't be true. And some of them are very good. They say, well, we got to learn. We have to know what you guys know. And I said, yeah, that's going to drop the cancer death rate. That's going to manage the disease. A lot of reasons for why what I said cannot be accepted. You have a multi-billion-dollar industry running on a somatic mutation theory of the disease, and it's not that. And yet everybody's making a lot of money on this. And then you come along and say it's a completely different disease than what you thought. Oh, how are we going to make these changes? But don't forget, we're not getting rid of standards of care. We're still keeping radiation, chemo, surgical mutilation. Surgery can be done as a final stage for the cure, because you can cure cancer with surgery. If you can take out we want to bring the tumor down, make it less angry, small, like a wart, something really small, less angry, less inflamed, and then you can surgically debulk it. So surgery is still part of this. Yes, we can still use radiation. Yes, we can still use chemotherapy. You just have to know the order in which you need to do these things, and that requires a retraining. So, and understanding what you need to shrink your tumor down, make it indolent, weak, and now come in with other therapies to potentially deliver a cure. Metabolic therapy is not likely to produce a cure, but it can certainly allow the combination of all of the therapies together to lead to a cure. So, but right now we're not doing it. We're not doing it correctly. We're using therapies up front based on a somatic mutation theory that's not effective. You have to rearrange the order in which you attack this tumor in the right way. You want to make sure there is no toxicity in the treated patient. They just don't know how to do what we already know how to do. And once our word gets out, you're going to see incredible responses on these patients. So is it going to be quick? No, it's not going to be quick. It could take you 6 months to a year to manage cancer in the correct way.
Yes.
But when you emerge from the treatment, you'll be much healthier than you were when you started. Not only will your cancer be under control, but your whole body will be healthier.
From your observation, when you target these cancer cells, do they disappear, shrink, slow down? What happens with these cancer cells?
We did this. So all three of what you said, they shrink, they die in different. You can see the dead cancer. We did that. You can look at dead cells under the microscope and see the dead cells. When we did glutamine targeting with Don, we saw massive numbers of dead cells, but we dead cancer cells, of course, in our preclinical system. And we saw some cells in other cases in humans where a lot of them were killed, but they were not all dead, and that's where you have to move the process forward to see if we can kill the rest of them. But here's the situation: if you kill a lot of cancer cells all at the same time, you have all these dead cancer cell bodies laying around in a particular tissue. Our immune system is designed to come in and gobble up and remove dead cells. That's part of what our macrophages do. Macrophage is a type of immune cell that will be effective in wound healing. So we kill a lot of cancer cells. The problem is if we are too aggressive on glutamine targeting, we prevent our immune cells from picking up the dead bodies, the dead cancer cells. That's why it's absolutely crucial that when you target glucose and glutamine and kill a lot of cancer cells, you have to pull the glutamine targeting off and let our immune cells get back to their normal job and picking up the dead bodies. If you're too aggressive on the glutamine, you kill a lot of cancer cells, but prevent the immune system from cleaning up the microenvironment, therefore leading to an infection and making another kind of a problem that you shouldn't have in the first place. Again, we have such a broad knowledge of cancer. We are able to track down those cells and kill them without harming the rest of the body when we know what to do and how to do it. Because I've looked at all these things. I have answers to your questions, mainly because I did the experiments next door with my colleagues. And we exactly do what you asked. What happens to these cells? Where do they go? What's going on with these things? Yes, we looked at them. And we saw exactly, some of them are shrunken. Some of them are outright killed. Some of them just hang on. We didn't kill them all. So that's why we're still perfecting the procedure. We already published the paper. We already set out the protocol, what to do and how to do it. And there it sits in the scientific literature right there. So you want to make it, read that paper, have the oncologists and physicians assistants and the whole operation follow what we are saying. You will drop death rates within the next several years. Here we did. We published. What more do we want? What more can I do? What more can we do? We already showed what's going on. We have some big papers. One of our papers just came out the other day. It's already got 16,000 views for the first time, showing that cancer cells are getting energy inside the mitochondria from a fermentation mechanism. The field did not know about this. That's why the glutamine has to be targeted. We have an outline. We have a clear outline supported by hard scientific evidence that we can drop the needle, move the needle significantly in dropping the death rate of cancer. The problem is that these are not the interference with what I'm saying has nothing to do with science. It has to do with policy and acceptance of the scientific facts, not the science itself. The science, to my mind, is quite solid, and it's based on decades of research.
Yeah. And are there ongoing clinical trials at the moment?
No, there are no clinical trials because I told you the hospitals think cancer is a genetic disease and they haven't figured out how to make a profit on that. Right now, the medical institution is not in the position where they want to change the standard of care. It's very, very profitable. People making a lot of money, hospitals, pharmaceutical companies, oncologists, they make a lot of money treating cancer. And now you come in and say, what are you going to do? Water only fasting and use repurposed drugs? Who's going to pay for this? You know, the only people to benefit from metabolic are the patients. The system is not yet ready to accept a therapeutic strategy that's effective and not toxic and cheap. You know, bottom line, it's a massive, expensive industry, and everybody is doing really well financially, except the patients. So, you know, you're not going to change until there has to be an outcry on the part of the world population to say, we want metabolic therapy. The problem is that the field doesn't, they're locked into an ideological dogma thinking it's a genetic disease. And therefore, the field all are trying to target genes. And then you have 1,700 people a day dying as the result of a misunderstanding of what the nature of the disease is. It's not complicated. When is it going to overtake? It should never, ever overtake heart disease, ever. We can drop those death rates by 30% in 5 years, 50% in 10 years. Once you start learning how to do metabolic therapy and running the clinics, that should be the way it should be done. Now, if people don't want to do that, so it has to go from policy. It has to go. Yeah, I just heard the other day I was saying, oh, we're going to make vaccines and we're going to do all this kind of crazy
stuff now they're talking about.
Yeah, we had Big Watson computer one time. He could beat anybody in chess. And he win all these games. So they brought him in to figure out cancerous, the gene mutations that might be causing cancer. And he was reaming smoke coming out of the machine and doing all this kind of stuff. And then he made recommendations to the oncologists. The oncologists were smart enough to know the recommendation from this super intelligent computer would have killed cancer patients. So no one told the machine that cancer is not a genetic disease. The goddamn machine was too stupid to know that. So AI is only as smart as the guys that are trying to work with it. So and then if you go on the web and say, ask AI, is cancer a genetic disease? Yes, cancer is a genetic disease. Then you go on to AI, is cancer a metabolic disease? Yes, cancer is a metabolic disease. Well, it can't be both at the same time. It's either one or the other. Is the Earth the center of the solar system, or is it the sun the center of the solar system? You ask AI would say, oh no, the Earth is the center of the solar system and the sun revolves around the Earth. And the AI, what about the sun? Oh, the sun is the center, and all the planets revolve around the sun. So I mean, again, it comes down to the theory. If the treatments are based on an incorrect theory, the outcome is not likely to be effective. And that's what we see, as we said. And right now, the pharmaceutical companies are gearing up that in 2050, 1 out of 2 people are going to have cancer. More and more young people are going to have cancer. The profits are going to be enormous in cancer. We can drop that. We can prevent all that stuff from happening right now, today. We have to do re-educate the oncology community to know that cancer is a metabolic disease driven by glucose and glutamine, and they can't transition over to ketones, so therefore transition the body to ketosis, target simultaneously glucose and glutamine, and bring in your standard of care procedures when you need them. And that's the solution to the cancer problem. And or a rational solution based on hard science.
And could you talk to us a little bit about hyperbaric oxygen therapy and cancer?
D'Agostino and I and others, you can't, if you bring your glucose ketone index down, you put a lot of pressure on the tumor cells, they'll produce ROS, reactive oxygen species from the high pressure oxygen. And then when they're in the low glucose and you give them the glutamine while they're in hyperbaric oxygen, it's a facilitator to kill cancer cells. It's a replacement of radiation and chemo in some ways. And it can make radiation and chemo even significantly more powerful by increasing reactive oxygen species, which will kill the cancer cell.
And how often is the glutamine therapy given? Is there like a duration and frequency?
That's the cutting edge right now. It's called dosage, timing, and scheduling. We are still working on that. And that's interesting because all of our funding for doing this is philanthropy and private foundations. Because figuring out dosage, timing, and scheduling is what they call, this is not sexy science. This is not cutting edge sexy science, but it'll keep damn people alive longer. That's what it will do. That has to be determined for humans. How long can we hit them with the glutamine targeting at what drug and what scheduling? And that has to be trial and error with each patient. And then once we see how patients respond, then we can implement it in a more outlined way based on our experience. But we don't know gender. You have issues of age and gender from people, and these all have to be worked out with dosage, timing, and scheduling. We just know what to do and generally how to do it, but the actual specific it's the glutamine predominantly because we don't have to worry about glucose. But it's that and what better drugs do we? How many different drugs will do the same thing? Targeting glutamines effectively without causing toxicity. What dosage? When do we schedule this? How is the timing? When the patient reaches a certain point, then we come in and do this. This has to still be worked out. And we do it preclinically first in the models that we developed. And then we get some idea. And then I translate it immediately over to the clinic, my physician friends, and they will say, we found out if you do it this way and that way, it'll better come back into the preclinical system and perfect. Feed forward, feedback until we perfect the system. You're not going to have everything right done. We have all the concepts down. We just don't have the actual the precision metabolism for each person.
You said, when the science comes, you cannot suppress the truth, and I'm documenting the evidence. And this is exactly what you're doing. So thank you very much once again. And I hope all of your hard work lead to the results and outcome that you are hoping for.
Certainly hope so. And thank you very much for asking really important questions that can help people understand what the issues are. And eventually, you're right, things will change for the better. And that's the hope that we have.